Hi and welcome to my OCR A-Level Biology Revision session with me, Christine. So today's lesson I want to look at osmoregulation, which is part of your excretion topic in Module 5. So the first thing we need to note is we need to look inside the brain, and there's a very specific area in the brain which is involved in osmoregulation. So osmoreceptors are found in the hypothalamus, and what they do is they monitor the inorganic ion concentration in the blood. So remember, your blood plasma is actually made up of water with lots of dissolved substances in it. So these inorganic ion concentrations are going to affect the osmotic potential. So this needs to be monitored. So the osmoreceptors in the hypothalamus are going to monitor this, and then what needs to happen is if there is an imbalance in our water potential, what we're going to do is the hypothalamus is going to produce a hormone called antidiuretic hormone. Now, the antidiuretic hormone, ADH, that's produced in the hypothalamus actually gets secreted into the posterior pituitary gland. It is stored there, and when it is needed, it is then released. So when we say that we have osmoreceptors that are monitoring the inorganic ion concentration, they are monitoring as to whether or not ADH needs to be released or less ADH needs to be released. So what does ADH do then? Well, ADH, the antidiuretic hormone, is a hormone which is a chemical messenger. It's going to be released from the posterior pituitary gland into the bloodstream, and then it will travel to its target organ. Now, the target cells it's looking for are the collecting ducts. When it gets the collecting duct, it's going to increase the permeability of the collecting duct. So let's just think about what other ways in which our body is going to monitor our water potential. Well, we actually have other receptors called baroreceptors, and again, you will learn about this in Module 5 Animal Response, but baroreceptors are actually looking at blood pressure. So when the aortic and carotid artery are being expanded because there is an increase in blood pressure, or if they don't expand as much because there's a low blood pressure, that is going to signal, it's going to be detected by the baroreceptors, and then it's going to signal via sensory neurons to the hypothalamus. Now, if there is a high pressure, that's going to lead to a reduction in antidiuretic hormone being secreted. So the key thing to remember is when you're looking at this, it's never one module on its own. It is always interlinking. But let's take a step back and actually look at this one step at a time. So we should have our correct water potential. If we have too little water or too many salts in our bloodstream, that is going to lower our water potential. If that lowers our water potential, our osmoreceptors inside our hypothalamus are going to be stimulated. We also stimulate our thirst receptors. And what's going to happen is the pituitary gland is going to be signaled to secrete more ADH, more antidiuretic hormone. That's going to result in the medulla absorbing more water from the collecting duct. Then what we're going to get is a small amount of concentrated urine being formed. So instead of the water going down the collecting duct to the pelvis and going therefore to the ureter and to the bladder, what's happening is the collecting duct has now become more permeable. Water is going to be reabsorbed back into the medulla of the kidney, and then it is going to result in a very small amount of concentrated urine being formed in the collecting duct. That should bring us back to the correct water potential, because the water in the medulla can then be reabsorbed by osmosis back into the bloodstream. And obviously, if our thirst receptors have been stimulated, we're going to feel thirsty. Water's likely to be drunk, which is a behavioral response. And again, that should bring our water potential back to the correct level. Well, what happens if we go the opposite way? What if we've had too much water or too little salt? Our water potential is going to be raised. Again, our osmoreceptors are this time not stimulated in the same way as they were when it was too little. They are going to not be stimulated, which is going to result in the pituitary gland secreting less ADH. So it's still our osmoreceptors, still located in the hypothalamus, but this time, instead of being stimulated, they are going to be inhibited. We're not going to stimulate our thirst receptors because we don't need to, and therefore our medulla is going to absorb less water from the collecting duct. We're therefore going to create copious dilute urine because we want to excrete the excess water. We want it to leave the body, because if we have too much water in our bloodstream, that water will move by osmosis into our tissue fluid, that will then move into our cells, and that could cause our cells to burst and therefore stop them from actually functioning. So we want to maintain this correct water potential. So the key thing to remember is, if there is less ADH being secreted, there will be less reabsorption, therefore a less concentrated urine, very dilute, and a large volume. If there is more ADH being secreted from the posterior pituitary gland, therefore there'll be more reabsorption from the collecting ducts back into the medulla. More concentrated urine is going to be produced, and that will be a small volume. So let's just look at how ADH actually works then. So the first thing we need to know is ADH is a first messenger. So antidiuretic hormone is the first messenger. It is a peptide hormone, so it's going to bind to receptors on the surface of the collecting duct cells. What that will then do is that will result in an enzyme called adenylate cyclase, which is going to be activated. Now, adenylate cyclase being activated is going to result in ATP being converted into cyclic AMP. So ATP is adenosine triphosphate. Cyclic AMP is cyclic adenosine monophosphate. So we still have our adenine, we have our ribose sugar, but this time we only have one phosphate attached, which is why it's called cyclic AMP. It's adenosine monophosphate. Now this is a secondary messenger. We are talking about one hormone, ADH, the primary messenger. We have now activated enzymes to create a secondary messenger inside the cell, because remember, if ADH is a peptide hormone, it cannot cross the phospholipid bilayer. So we need a secondary messenger inside to then activate other proteins. So this is going to activate protein kinases. And when you do module 6, you're going to learn about post-translational modification. This is an example of post-translational modification, where cyclic AMP is going to activate protein kinases. And these protein kinases are going to cause vesicles that contain these aquaporins. These are channels which allow water to move. So these aquaporins are attached to the vesicles. Remember, a vesicle is a biological membrane. The aquaporin is a protein, a channel that is going to allow for the water to move. That is going to fuse, because it's made of the same stuff, it will fuse with the collecting duct membrane, and that will therefore increase that permeability of the membrane. So if that increases the permeability of the membrane, that means that more water will be able to be taken out of the nephric filtrate, taken back into the collecting duct cells, into the medulla, and then into the bloodstream. So it's important that we remember the function of the kidneys. We have the renal artery, which is providing this unfiltered blood. We have our afferent arteriole with our high blood pressure. We have our efferent arteriole with our lower pressure. And that results in ultrafiltration occurring at the Bowman's capsule and the glomerulus. That results in filtered blood and our filtrate. Now, the key thing to remember, as the filtrate makes its way through the kidney nephron, after it's left the Bowman's capsule, it will go to the proximal convoluted tubule, the PCT, where selective reabsorption is going to occur. That's where your glucose, amino acids, hormones, vitamins, water and sodium ions are selectively reabsorbed back into that filtered blood which is circulating around the kidney nephron. At the lip of Henle, again, selective reabsorption. This water and sodium ions. The water and sodium ions are going to be moving into the medulla, and then they will be moving, if necessary, into the blood to ensure we have an isotonic point. We then move on to our distal convoluted tubule. Again, selective reabsorption, the water, the sodium ions. And remember, when we talk about the distal convoluted tubule, we're going to bring in our aldosterone hormone, which is going to either activate the reabsorption of the mineral ions, or if there's less aldosterone, there will be less reabsorption. We end up at the collecting duct. Again, this will be regulated with a negative feedback of our ADH hormone. So we've got selective reabsorption again. We have our water and our sodium ions. So what we result in is regulated blood leaving the kidneys via the renal vein and urine being produced and sent down through from the pelvis, through the ureter to the bladder to be excreted. The urea, remember, is that nitrogenous waste that the body needs to get rid of. So urine is made up of a waste molecule and also excess water and excess salts. It should not contain any glucose. It should not contain any blood. It should not contain any large proteins if the kidneys are functioning properly. So I hope you have liked my video, and if you have, then please do click on the like button and subscribe to my channel. Also, if you haven't done so already, please do check out my revision platform, www.aiqchat.com.